Hello guys. So yesterday I have posted a question on my Instagram that what is the length of actual human DNA? And so many of them were correct, but some was wrong as well. So the actual length of a human DNA in a particular cell is 2.2 meters. And why it is so? Let's find out. It's a very cool concept, very different concept. Some of them must have heard it somewhere. The DNA is the genetic material present in the nucleus of every eukaryotic cell, and the length of DNA varies from species to species. The length of DNA can vary a lot. In humans, there are specific nitrogenous bases, and each nitrogenous basis has a specific gap between them. Nitrogenous bases, you can represent nitrogenous bases as adenine, guanine, cytosine, thymine in DNA. And out of these two, the black one, which I've wrote, adenine and guanine, these are called as purines. We call them as purines. They have a large molecular size, two-chain structure, purine chain, and they are cytosine and thymine, which comes under pyrimidine. So there are different theories, and they are all responsible for the actual shape and the structure and size of DNA. And there are so many different things. First, the DNA has two strands. One can be called it as a parental strand or father strand, or next is maternal strand, paternal and maternal strands. You can call them as, and both of them have antipolarity. Suppose a strand is having 5 prime to 3 prime direction, another strand will going to have a polarity of 3 prime to 5 prime. Different, there's antipolarity between anti-parallel strands, you can say. And another thing, between both the strands, there is a specific distance of 2 nanometer. There were four scientists, Watson, Crick, Morris Wilkins, and Rosalind Franklin. They all determined the whole structure of DNA and the actual size of DNA. So the diameter is of 2 nanometer. It is fixed. It is fixed. Next thing is that the backbone is made of phosphate, phosphodiester bond, we say it as. And between two nitrogenous bases, suppose I have chosen this example, if the distance between this C and G, cytosine and guanine, is fixed, the distance between two nitrogenous bases is 0.34 nanometers. It is fixed, 0.34 nanometers. And now we can calculate 0.34 nanometers. It is fixed. One thing is fixed for humans. Humans has total 6.6 into 10 to the power minus 9 base pairs, or we can call it as nitrogenous bases. So it is fixed for humans. Modern Homo sapiens, it is fixed. Total nitrogenous bases in a diploid cell, okay? In diploid cell. Now, how can we calculate it? We know the distance between each and every nitrogenous base. It is 0.34 nanometer. If we multiply 0.34 nanometers, which is fixed distance between two nitrogenous bases, and with the number of bases in humans, if we multiply both of them, it comes 2.24 meters. And it is approximately 2.2 meters, you can say. So the correct answer is this, 2.2 meters. Why? Because we already know the distance between two nitrogenous bases. There are 6.6 into 10 to the power minus 9. It comes about 6.6 billion bases in a one DNA, billion. And the actual distance between each nitrogen with 0.34 nanometer. There are two strands. Both the strands are antiparallel to each other. One I have represented through red color, another through black color. They both have polarities on a different side. The nitrogenous bases are of two different types, adenine and guanine. They both have their chemical structures. I'll be telling you all these different structures in much more detail. In further details, like adenine has why adenine is different from guanine. If both are purines, they must have a different specific groups, like 6 amino purine. If amino group is present on the sixth part of a chain, then it will be called as adenine, and guanine has a certain different structure. And same goes with cytosine and thymine. There are different molecular structures, both of them. Adenine and thymine form two hydrogen bonds together. There is another one, Chargaff rule. Chargaff rule is very famous in molecular biology. And according to Chargaff rule, adenine make two hydrogen bonds. Both the strands are attached together by hydrogen bonds. Both the strands are attached together by hydrogen bonds. So adenine form two hydrogen bonds with thymine. And on the other hand, the cytosine form triple hydrogen bonds with guanine. This is another big concept. Like the number of purines and the number of pyrimides remain same from species to species. Number of purines, A plus T, it also gives like number of purines, A plus G. Purines must be equal to C plus T. A plus G is equal to C plus T. And this will be constant. This is very much required in all of the numericals, basic numericals of molecular genetics. And this was a concept. I hope you like it. And if you have another such queries, I'll be posting you on this YouTube channel only.